I'm not saying because that could get me in trouble, so I was saying maybe.
Within reason.
Yes. I always obey my speed limit. Seems like there's more knocked over C-pens than live C-pens.
Abby's saying linear growth rates in a boot sponge were about 2 centimeters per year. So a 30 centimeter tall sponge is approximately 15 years old.
Wow.
Thanks, Abby.
I, for one, am really glad your internet came back.
So am I. So I'll be one. I'll be two.
Although Starbucks would be nice about now.
If you like that sort of thing.
Yeah, we're starting to go over the darker backscatter return. I'm not seeing much of a difference.
Nope.
Maybe affected by the beam angle on the slope too. Something.
That rock.
There we go. That caused the whole return.
Yes, Abby, you can go walk the dog and come back. That's Ferrea too. Dead.
Dead, yeah. Like, I wonder if the associates when it were alive were very different.
Yeah, that's true, because mostly when you see them alive, it's brittle stars and not much else. Are we getting urchins now?
Are these baby urchins?
Pretty small.
Need to tell them about the kelp holdfast we saw back a bit.
They're on a trek there.
Yeah.
It's going to take them a while.
It can be a problem for kelp forests. Is that true? With like it's like the sea otter population in decline brings the urchins to be a problem.
Yeah, we've been dealing with them in Monterey for the past 5 years, 4 years. We had the sea star wasting disease that killed a bunch of sea stars. And then we had a great recruitment of sea urchins. And then the next year there was like no kelp.
Oh, man. All right.
And it's slowly coming back. They're actually doing that with the Fish and Wildlife Service. They're setting out recreational divers are petitioning certain reefs to go out and clear all the urchins off, or lots of them, and see if the kelp will come back. So they're doing one off of Northern California, and they're actually petitioning one right now to do in Monterey Bay.
That's awesome. We're actually
Yeah.
right now at the end of the transect, so we can all mark it.
Roger that. Thank you. And we can just keep going.
All right.
Nothing to see.
Bridge. Yeah, it would be neat if that really makes an impact. Like I know in Florida they were doing the lionfish smash and, you know, incentivizing people to go out and kind of cull some of the lionfish that were invasive and problematic. I don't really know, like I have no understanding of the efficacy of that. I mean, I can't imagine, I mean, you know, I know it will help, I just don't know how much of a meaningful difference those things can bring.
Well, they have no natural predators, so if people spear them, which is one of the things they do, and then they eat them. They're actually not bad eating. You just have to be really careful around the spines.
Yeah.
So it's like you don't carry your bag on your side once you've speared it, because if the bag washes into you and the spine goes into you, that's not good.
Ow. Yeah.
So definitely precautions to take.
I've seen they make their bags out of a piece of PVC piping.
Just for that.
So the spines can't stab you.
That's a good safety feature.
And I've also read that the culls aren't doing much, but they're increasing awareness and just letting people know not to dump their aquariums into the ocean.
Oh, is that really the source of that?
They think so.
Wow, that's sad. Totally avoidable.
Oh, we have a snailfish coming up, if we want to zoom on it to the right.
We're still going 0.4.
Reg.
Do you want to?
Yeah, how much longer do we have on the sandy flats? Yeah, 0.5 would be good then. All right, Marley, go ahead.
So is there that much of a current that it's holding station?
Yeah.
It's like, leave me alone.
Hard.
Making some headway there. All right, come with there, Marley. We'll leave him alone.
Oh, and it has a caudal fin, so it's not ophididae.
There you go. That's a black-tailed snailfish. It's pretty distinctive.
They have the prettiest blue eyes.
Oh man, I didn't see them. Wasn't paying attention.
Let's go back.
Yep. Cuteness check.
I don't know, you think hagfish are cute, so it's like, you know, it's a big check.
Yeah, it might be too cute for me, you know.
Some more of this spongery here, wow.
Yeah.
Again.
And one time there was a huge reef here. Back when the dinosaurs were here.
Did you guys want us to slow our features like this or just keep going at 0.5 knots?
I think we can just go. Let's make some headway.
Reg.
We've already looked at a lot. Unless we see something new and different in here, but so far I don't see anything different.
If this corresponds to the backscatter at all, it's going to be another 100 meters. What's up?
Reg.
Wow, another reef. Here comes a granddeer.
Tom, how many years have you been working on Piggy Bank?
Piggy bank specifically, the first time I ever worked on it was 2010, but our group has been working on it since about '02. I think it's probably the first dives we made out there. So 18 years.
That
And
must
this just isn't an area that anybody's been to yet, huh?
No, because the top is like 280 meters. So, you know, unless somebody's got some money to plunk down to go survey deep, nobody seems to really care about these deeper areas. I mean, they are now. They're starting to realize it, but when we were first doing this, everybody wanted to know basically the rockfish. Well, what's going on with the rockfish species, which is extremely important because we want to manage the fisheries. But then we found places like this where you have all these sponges, and now we see these sponge reefs that we have here. They're dead, but at one point we had sponge reefs here. So it's just interesting what you can find in the deep sea, especially when we're, just tens of miles away from suburbia on the coast.
Yeah, it must be weird to study for something for so long and then only recently have access to such a big and different part of it.
Yeah, it's true. For years, we only used the Delta submersible that went to 365 meters. So for us to look deeper than that, it was really rare. And then we got involved with Mbari, and they can go pretty deep. Got involved with other submersibles that could go to 600 meters, and then other ROVs that went to 600, and then you guys coming out here that could go nice and deep. I mean, similar to Mbari stuff, which is the Monterey Bay Aquarium Research Institute. Since I'm just using an acronym, I got to be good. Not to be confused with the Monterey Bay Aquarium. They are sister groups. They're not the same.
Wow.
I would almost say we could do a mosaic here, but there's really nothing that would get us. You need some hard, specific target in it to come back to. And I don't see anything that we'd be able to pick out.
So the habitat photo mosaics are intended to be something that you can come back on and compare?
Yeah, basically, and the 3D ones that we did with the rock with the Peristanella coral on it yesterday, and the Lophelia one, we can come back and compare those over the years. Like the Lophelia one will be great because it's one of those things that unless you go down and mark it, you don't know how much that Lophelia has grown, because we were talking about that. Right. It's just kind of there, and you see all the rubble, but is that because it's rubble from 100 years, or did a whole bunch of stuff fall off and die recently?
Right.
So, yeah, the Paragorgias are flapping in the current, so there must be a ripping current here.
Dover.
By the way, there's a bunch of sponges too that were so happy.
For a while.
There's a hagfish inside.
Maybe.
And back to the mud.
It's very periodic.
Yeah, I'm trying to mark it, but it's like there's a beginning and an end, and then resumes real quick. The sea pens here aren't dead.
Oh,
Or
good
some of them
observation.
are dead.
Now I see a couple in there now, yeah. But I see more uprights than I did the other place. But at that last place, there was a lot of bedrock. So maybe it's just bedrock outcrops around here got all this Pharea and then something caused it to die.
What type of sea pens are these?
Oh, look at these.
I'm thinking that some of these taller ones might be Holypterus.
Like running
I
through
was
the
instructed
Sorry, go ahead.
the other night that the deep ones were red and the shallower ones were white. So I would concur with the Holypterus deep red identification.
Deep red confirmed.
It's very cool.
Oh, wow.
So as Lizzie's saying, if we come across another bioherm like that, we can find a spot that maybe we can do a 360 if there's a particular point where it comes up. We can do a 360 mosaic there.
Roger.
Sure. Of one of the sponge reef outcrop things.
Yes, correct.
Roger that.
And Lizzie's question is, why did we go anywhere else? I should have just stayed here for 4.5 days. And now we found a huge field. Can we just zoom in on one of these if you
Yeah.
get a chance?
Of course, yeah, for sure. I'll go to this little
And
poster.
that way, yeah, that way we'll be able to confirm.
All right, Marley, go ahead.
Well, Meredith will be able to confirm.
That's great. You know, if you want me to just sit down and get tied on one.
Actually, this would probably work good. She can see, and they're basically all the same. They're on 2 sides, maybe on 3 sides, but they're one side's not.
Oh,
I think we're good. Thank you.
Sure.
are the polyps all oriented the same way, or is it just how the current is?
The current.
I think the current's blowing in that way.
Yeah.
You can see it also on the starboard there. They're all blown. See
Oh,
all
yeah.
blown over there? It's like an isometric perspective.
They were probably knocked over from the giant squid that came through.
Obviously.
Just missed him.
Right over there.
You guys don't want to transect through some of this stuff, do you?
Do you want to count this stuff?
Once we get 100 meters, we will, but
Yeah, we're over 100 meters.
and how far do we have left to the next point?
So we have just under 200 meters. But
All right.
yeah, it wouldn't take us
Yeah,
too far.
let's start a transect.
Let me just double check that I don't want to. Yeah, let's keep 065.
You want to slow the ship to 0.4?
I will.
They are going to hate me.
Yeah.
Guess
I
it's
can
job.
see it.
It's job security. That's what it is.
What do you think? They'll have more difficulty with the field
then.
of sponges
So you're
on
ready?
Pharea or these sea pens?
Mark it.
Roger that.
I
Stepping
think
forward.
they'll have much more difficulty with the sponge fields because there's
One.
nothing else but mud and this.
Brittle stars.
Yeah. And a couple thorny heads.
Become a sea pen expert for one species.
Well, that's the other thing, is to look to see if anything looks different, but you just assume that right now, if they're all the same, you don't see any different color. Like, they're all pretty much the same height. There's a few little ones mixed in, but
Yeah.
they all have the same kind of look.
Yeah, that's a real biasing challenge for me when I've done imaging stuff. It's like you get so used to one form of this thing that you're so used to lumping everything into it. Not that that's usually not fair, but
Right. I mean, that happens with fish. It happens with anything.
yeah.
You start counting fish and you just start, oh, that's a school of this, as opposed to really looking through it and saying, oh, but there's 5 of these other things mixed in.
Right.
Yeah.
These thorny heads think they're in a forest.
Yeah, that would be an interesting perspective. The ophiuroids are like birds to them.
They're like flying squirrels. They jump off when there's a big current, then they catch another one.
These are just startled squirrels.
Oh, we have to relate it back to the squirrel tails. So.
Right. Another sea pen though, right?
These are squirrel toothpicks.
Squirrel trees.
They're squirrels after hibernation.
Yeah, stretching.
Squirrel Charlie Brown tree. The diversity and habitat on this dive is just so interesting. And from way back when they just had a little bit, then we hit the sponge reef, then we hit the area with dead sea pens. Now we've hit the one with just tons of sea pens.
Yeah, and they've been like reoccurring too, some of these.
Yeah.
Unfortunately, it's not just the backscatter that's correlating. You have to figure something else out.
Well, that's why you come in ground truth too.
Yeah. Well, it's probably something more interesting than just that.
I mean, I could see that the backscatter could give you some hard return if it was hitting off sea pens and going up.
Yeah. And even like, you know, if you have sediments that are the same size, but some are more compacted or, oh, I don't know. Oh,
Yeah, we were looking years ago off of Davenport, which is kind of north of Monterey Bay, at these supposedly hard areas off of just before the shelf went down into the slope at like 100 meters. And we went there to look, and it was basically these giant beds of brachiopods.
oh wow.
And, you know, they were tens of meters across. Just, I would say there were, millions of them in these areas. And the geologist who's with us was saying that he thought that the brachiopods were living on the brachiopods and attaching to more brachiopods and basically making a brachiopod reef over time. And that he didn't know how deep it was there. But his theory was the bottom at some point was hard. They started living, and then the sediment would come, and the sediment would shift over time. So instead of having just one big area, you had these different areas because something about they would create their own, the sediment would go around them,
Yeah, like
and kind of not their own scour, but it's something like that.
Wow.
And he had this whole theory, and so that's why they could persist out in the mud now.
super interesting.
Yeah.
Biology can beat geology.
That's why it's been interesting the last few years. We have this campaign on the West Coast called Express for expanding Pacific research in submerged systems, something like that. But it's basically just a group of scientists from different disciplines that get together. So we have folks from NOAA, so Erica and I, from Northwest and Southwest Center from the West Coast, then from NOAA from the sanctuaries. Most of them are involved, most of the people from the sanctuaries. Then USGS, and then the Bureau of Ocean Energy Management, BOEM, and MBARI is involved. And we go out on cruises and we share time on the boats and expertise on the boats. So we would have geologists on our cruise. They would have fisheries people on theirs or sponge people. And that way it really leverages the time and the expertise among all the groups to be able to get some of that kind of information. Like, hey, if you look at the geology, it makes sense.
Yeah,
Something I wouldn't have thought of.
yeah, it's super neat.
Yeah.
I think that's a big part of why I like this field is every question kind of impinges on each other. Like no field is isolated. It's all very interdisciplinary.
Totally.
Yeah.
Because you could settle someplace on one rocky area or one muddy area, like we've looked at, and then you can settle in another one, but there's nothing, no food. The currents aren't good, so you don't get any food.
Yeah.
Or why are all these here versus the other place they were all knocked over? Besides the giant squid theory.
Which almost always holds, I assume.
It does. They get around. There's the snailfish behind that crab.
Yeah, the little
Were
guy.
you talking about that the other day, Erica? There was a blacktail snailfish behind the tanner crab.
Yeah, the snailfish actually will lay their eggs inside the crab's carapace. And the crab will carry around snailfish eggs.
Wow. Oh, and it actually injects inside the carapace? That's what you're saying?
Yep.
Oh, wow.
So up in the gill cavity of the crab, you'll find these batches of snailfish eggs. And we did that project for a couple of years on the trawl survey, and it was more than one species of snailfish that did this.
Does that kill the crab?
We don't know. It didn't seem to bother them. It was just kind of a protective space. But we just were able to confirm that they were there and that they were snailfish. And I think that's all we were able to do as far as that project was concerned. So if somebody wants to try to watch the whole cycle with a bunch of captive tanner crab, that would be a cool master's degree.
Yeah. Rate your
So
level
all
of
you master's
discomfort.
students out there. Erica just gave you a free project.
I have no funding. It's just an idea.
But you get to eat your subjects when you're dead, when they're dead at the end.
They're great with melted butter.
How many scientists could say that?
Definitely not sponge people, I think.
They clean up their plates after with them, though.
Oh dear.
That's that interdisciplinary work again.
Right, right.
So there's actually another smaller red sea pen in here. I think, is that one the funiculina, Tom?
Yeah, I think so. I see one right there that it looks a little thicker
Yeah.
and darker color. Good eye. I wasn't paying that much attention. That's why you're doing it. You got those young eyes.
Your analysts are not going to be happy.
Just be careful. There might be
Because-
another time.
Well, there's umbellula in there too.
And there was at least one anthoptylum. There was one squirrel tail in there.
Oh,
I
man.
did see that one, yeah.
Of course you did. Well, there'll truly be a sea pen expert by the end of this translate.
Yeah, a stick in the ground is a sea pen. There you go. You're an expert.
Hey, I imagine there's new art.
Yeah, when you broke to go eat dinner, a long spine or a long nose skate came through.
Oh yeah.
And I was like, oh, you could have got that.
Here we're not allowed to take on pets on the ship though, so I think a long nose would be out of the question.
Maybe we could fit it in the wet lab. Get a big enough tank.
Just put it in a tote on deck.
Sure, would be super happy to be our pet like that.
I think we get bad karma for years if we did that.
Oh. I had a friend in the Bahamas who took a nurse shark. Well, he was teaching a course there. And he took a nurse shark and just, yeah, had it in like a, what do you call it, like a cooler for like a day or two. And, you know, was saying that, you know, he has a ton of experience and that they're totally fine. But like, I was so unhappy about it. I didn't enjoy the experience. As someone who likes nurse sharks, I wanted to like interact with it and everyone was happy to, but I was like, this is so upsetting. But I guess they're fine doing that. To have a little senescent couple days, but.
Interesting.
Depending on your definition of fine.
Yeah, I know, right? Glad I'm not the only one. Nurse shark.
Yeah, well.
Nurse shark.
You did say nurse shark, yes.
Oh, okay.
So one of my jobs is I scuba dive and count baby rockfish. But then I also spear the baby rockfish for aging. So.
It's a complex relationship.
It is. I kill 6-month-old baby rockfish. And it's like, well. So I can't complain about somebody putting a nurse shark in a tank for a little while.
Well, it was fine. They did teach me you can just grab them by the tails.
Yeah, don't grab them by the other end.
Yeah, exactly. Yeah, I had a friend who was doing, like, I forget the reef fish, but, you know, she watched and monitored these individual fish for many years, and when it was time to collect this one, she lost the bag, you know, and felt so bad that she wasted this fish, you know. Just so many years of observing it and finally having to take it.
That is true. I know that my thesis professor.
Oh, and we can
Did
mark
a
the
tagging
transect
study.
right now. Sorry.
Roger that.
Rog.
He did a tagging study on these for home ranges, and at the end he had to kill them. It was just like, oh no. I've been watching them for 2 years.
Yeah. It's like, oh wow, they're all primed for success. Let's take them. Yeah, so I'm going to change our bearing slightly for that ED target. Still not going to be much change for a little bit. But I don't see much to maximize on the way there.
Sure.
Yes,
So
it's going to be 077, so let's go 080.
Rog. 080.
Thanks.
And what's the distance to ED?
Yeah, it's like almost a kilometer. It's 870 meters.
So
away.
we can sprint if we
Okay.
want,
Yeah, I can
because
bump
I
this
think
up
it's
to
going to be
0.5.
like this for a while.
Yeah, I agree.
0.5.
I'm waiting to mark when these C-pens aren't around, but maybe they'll just be on this whole thing.
Yeah, before we take off, could we zoom in on one of these really small, just a snap zoom on one of these small, more reddish C-pens?
Oh, the
Trying
kind
to see
of
if one's in the frame.
short ones. I think
There's
I know.
one to the left.
Below the laters.
No, even farther over. But yeah, now it's below the lasers.
Ooh, nice.
Oh, we got one each in there. Quick, oh no.
How would I drive up to these ones up here?
I can stop the ship for a second if you
No, no worries.
Okay.
These are also the same ones.
Yeah, those ones to the left totally are.
Marley, go ahead.
They all went to different barbers.
Sorry, there's going to be a bit of a sediment cloud from my abrupt landing. Come wide, Marley. Find one that's out of the sediment cloud. Right. Yeah, that's a good point. You know what? Running this one. All right, Marley, go ahead.
That is awesome because we get both of them in there. Thank you, that's good.
All right, full wide, Marley.
Call.
All right.
Are those tanner crabs that we're seeing? Do they bury themselves like that, or was it dead? I don't know if you said.
I actually didn't catch that one. I have not seen them. Cancer crabs bury themselves, so it's possible.
Yeah.
But the cancer crabs are kind of fun because you go by and you just see the eyes and the mouth parts. Everything else is underneath.
Crocodiles. Like, I think it was
There's
that one on the left, the same species.
Yeah, that's the tanner crab, the groove tanner crab.
Maybe the other one was
There's
just...
squirrel tail. See some more stars here, though. Now they're gone after I say that, but I was seeing them.
You'd think the hagfish would want a place with more hidey holes.
Yeah, they definitely, I don't know, around 100, 200 meters in Monterey Bay, they definitely are in the mud. There's just tons of hagfish holes everywhere.
Wow. Yeah, and we're just, we're like 95 meters away from the last transect, so if you want to grab another one on the way.
Well, let's wait like another 100 meters. You said we had close to a kilometer, so we can
Yeah.
put a little bit more distance between us.
Roger.
See if something changes.
Cool.
Yeah, if we can close in toward ED, I think that'll give them 8 hours to try to do a photomosaic and find sponges in that area.
Should be good. Yeah, those photomosaics, the habitat ones, the 10 by 10, they can take quite a bit, it looks like.
Yeah.
Especially because we don't have real downward looking cameras for that, so we kind of want to get it from the two headings.
Yeah, we were talking about that at lunch and maybe doing more of the smaller, like the rock that we did for Lophelia or for the Peristanella coral.
Yeah,
One
that's
of those.
convenient.
Because those you can come back to and really study. If we just grab a habitat one, it's hard to come back to the exact same spot if we don't have some kind of marker in the bottom.
Right, I see.
But the one with the Acanthogorgia, the yellow coral, that one was pretty cool looking area for that. For the habitat one. If you guys saw that, it wasn't on our watch. Do you guys watch the video and stuff when you're not on watch? When you're awake, not on watch?
Oh, yeah.
Yeah.
Okay, just wondering. Is it all throughout the ship? Do they have different monitors?
Yeah, there's a bunch of monitors throughout the ship.
Yeah, you can watch it from a much more relaxed perspective.
This is very relaxing though right now.
No, this
The
is not.
sea pens and the fish going.
No, it's always fun.
Do you grade the other watches and like, oh, I wouldn't have done it that way. What are we doing?
No comment.
Everyone does it their own way. Probably say the same for every watch.
Yeah. What is interesting
Yeah.
to like work with different pilots. Each one does have a different style, and not necessarily, Yeah, I mean, you can tell who's piloting sometimes. They're all super skilled.
Yeah.
It's just a different manner of kind of how they fly around and what their
Yep.
approaches are.
That's a good idea of keeping you guys in the same teams that way so you get used to each other.
Yeah.
Just as clearly the most skilled, but
Yes.
Oh.
she's super good.
This is the A team and the fun team. So
Fun team will take. Yeah.
yeah, I haven't given you a hard collection yet. I'm gonna have to work on that.
I know. A little behind
Can
on
you
your game
reach-
there, Tom.
Yeah, I want you to reach under the sediment and grab me a knocked over sea pen that we can't see. Just do a blind grab someplace.
There'll be a lot of holes in the ground around here. Like, nope, hagfish. Next one.
There's one. If you can grab that hagfish, that's extra credit too.
Get Ben a pet hagfish.
Yeah. My own feather boa.
If you slurp a hagfish, I'd give you extra credit because once he comes up, you're going to be not happy.
We'll just have a gelatinous cube.
Just let Ben deal with that one. Like, none of us data science people on board will touch it.
I'll deal with that.
Yeah, I've always wanted to hold
Wait.
a hagfish.
I know, right?
Wow.
You could keep one of those in a cooler.
A hagfish in a cooler.
But how bad would I feel? Pretty bad.
No, you wouldn't because you'd name him and you'd feed him and
Right,
you'd be all happy.
Like, I can't go to watch.
You
I have to play with Sam.
You can name him Haggis.
Little Haggy.
Sounds like a Viking hagfish.
You can get him a little hat, a little Viking
Ugh.
helmet.
My heart is full.
And if he ever died, you could give him a Viking funeral. All the fun you could do. I think that's going to be the next Christmas major present. Hagfish in a box. Just mail it to everybody.
I'll put it on my Christmas list.
I'm
It's
gonna
just
write,
a
I'm gonna
daily
write a
suck-
hagfish Christmas story. Send it to everybody.
But you can't read it. You just have to put it in Ben's stocking. That's all.
Yes.
Should make it a children's book and try and get it published.
It'll be a carol.
Okay. The snow is slime.
Jingle hagfish, jingle all the way.
There we go. Marley, first sentence written. My cat bites me all the time. It's okay. I'm used to it. I'm like, oh, it's just how she shows me she loves me.
Oh, the hagfish slime me. Just shows you
Yeah,
how he loves me.
exactly. Oh, it's not defense. It's just affection.
Isn't the hagfish slime also used for medical purposes? That there's
I
like
know it's
some
like one protein that is like so efficient per volume of water that it gelatinifies. I don't know. I don't know if you know any more about their application, Tom.
No, I remember hearing that
I'm
a
actually,
truck overturned with them in it.
yes, that was up in Oregon.
Yeah.
What was up in
But
it?
I'm looking online, and it says the synthetic hagfish slime may be used for ballistics protection, firefighting, anti-fouling, diver protection, or anti-shark spray.
Anti-shark spray.
This is practical uses of hagfish slime. So I don't know.
It's much more aggressive
That's
uses.
the first thing that came up.
Wow.
But yeah, up in Oregon, there was a truck that was driving that was carrying hagfish and got in an accident and sloshed it across the freeway. And they basically had to spray it down because all the cars got in accidents because it was so slippery on the road.
Slip and slide.
Is that real life?
I think Di actually put that in the chat a while ago, so that, because I saw the hagfish report, she put I think it was her, put that in, so it's off my screen now, but it could be back there. I mean, I could probably find it.
I think it was in the chat a little bit back.
But that was a synthetic, or was that actual?
I thought
This one said actual, but then it was highway. The other one was real hagfish
Wow.
that dumped on the freeway.
Oh my God. I'd be like, that's highway hallucinosis I'm seeing. What's going on?
Yeah, it was in
Yeah, you're right.
2017 in Depot Bay. Apocalyptic hagfish spill covers highway in slime.
You sure that's not the Onion?
Yeah. I just put it in
National
the chat, so if
Geographic.
any of you have, if any of you have a way to grab it out of the chat right now, you could look.
On it.
There's some grody pictures.
Oh man.
Can you post those pictures on channel 3 and show everybody?
I'll start crying.
If only.
Too cute. Too sad.
Oh, there's some non-happy pictures in that. It might change your mind.
Oh, wow.
The rasping jaws. Oh, wow.
Just the oh wow gives you the clue right
I
there.
mean, if I'm looking at the one Erica just posted in the chat, too. That car.
Well, I wouldn't think the slime would be the worst part. It would be picking up all of the hagfish that were squiggling all over the road.
Poor thing.
Poor thing.
Yeah, so I think we've gone far enough. We could probably do a transect now.
Yeah,
Roger.
we're still 600 meters from any change, so, you know, maybe,
Right.
maybe
so we'll
there's
just
something.
do one. I just didn't.
Yeah,
I figured we'd get some distance before we did it.
No, that's good. And there's asteroids now, so something's happening.
Yeah.
All right, you good to mark it?
Sure. Tom, did you want this at 0.5 or?
Yeah, or to bring it down.
Yeah, that's fine.
0.5?
Okay.
Raj. All right. I'm ready for you there, Ben.
Okay, marked.
Raj. Stepping forward.
Yeah, if something comes up, you have permission to slow down.
Okay.
Roger that.
Not like you need my permission if something comes up, but oh, there's a line, but Tom didn't say I could slow down yet, so I'm just going to keep going.
How we roll.
Gotta get the transect in.
Yeah. What is it? Urchins now? What? I looked down for 1 second.
I know. Trying to mark the start of a transect and suddenly urchins appear.
So that asteroid is on the left. It's like center portion. Is it just raised like that, or? Yeah, no, that's just the morphology. It's not like enveloping
Right, that's a
something.
cushion star.
Yeah.
Because here's another one right
No,
here.
it's yeah, the hipposteria. And they can change their water pressure and
Oh.
how fat they are.
I wish I could do that. I had a water vascular
Right?
system like that. Marley, look at me. No peas. Wow.
With the lack of sea pens or less than what we've been seeing, have anything to do with how many urchins there are now?
It may. I can't say. I don't think the urchins are eating the sea pens, but maybe they're competing for space or knock them over if there's so many of them. Oh, you know what it could be? They also can contract into the sediment, so the urchins are disturbing them and then they withdraw to the sediment.
Huh. That's interesting.
That's a cool superpower.
It is.
A little blip on the amazing. Here I can show you. Oh. Hmm.
Yeah.
Yeah. Oh well.
30 meters over.
Nope, no. What is that?
It was a random chunk of area out in the middle of nowhere.
Yeah. I don't see anything else like it.
Was it a dead one? I didn't see.
No, it was alive.
Oh. This outsider did well.
It crawled away.
Oh God. Carnage.
Long nose skate to the side.
Good eye.
Front.
So much for the urchin idea of having them contract, because now they're just gone.
I wonder how much of it is just totally random recruitment success on these flat plains.
Could be. Because there's been, like yesterday when we went across the flats, I don't remember seeing any, and we were, what, I guess less than a kilometer away on the mud flats.
Yeah.
It's another sponge.
That one has seen better days, though.